Welcome to my video on dihybrid crosses. If you are still not sure what a monohybrid cross is, then you probably should view the monohybrid video first.
A dihybrid cross is one where we cross birds that are heterozygous for two particular characters. We can use a modified Punnett square to work out the ratios of genotypes and phenotypes expected from such a cross. So let's get straight to an example. Let's say that Mary has an unrelated cockerel and hens who are both heterozygous for Colombian, and they are also heterozygous for blue egg colour. Although hens lay eggs, the cockerels also have these genes and pass them on to their daughters and their sons too. It is worth noting here, if you decide to breed for egg characteristics, for example for dark brown eggs or large eggs for winter production, then the male line is as important as the female line. Mary wants to know what genotypes and phenotypes might occur among her chicks if she mates the heterozygous hens together with a heterozygous cockerel. How likely, for example, is she to get Colombian chicks that lay blue eggs? To find out, she constructs a Punnett square to help her understand what could happen. This is how she does it. First of all, she draws a 5x5 box to make her Punnett square. Then she divides the first top left side box diagonally and writes cockerel and hen as shown in the figure. She shows the genetic contributions from the cockerel along the top row. Each of the 4 possible allele combinations occurs with a 25% chance. The filling in of the boxes needs to be done methodically to avoid confusion later on. Down the first column, she shows the genetic contributions from her hens. Once again, each of the 4 possible allele combinations occurs with a 25% chance. She starts at the top of the box with both of the dominant alleles first, working down the boxes so that both of the recessive alleles are last. Again, it is important to be methodical, especially when, as in the case here, the capital and lowercase letters look very similar. Exaggerate the differences if you need to. It is helpful to realise that if this was a different cross, the hen and the cockerel contributions might be different from one another, and then the reminder in the top left box would be particularly useful. Now, she fills in each unfilled box with the possible genotypes of the chicks. These will be a combination of what the cockerel and the hen donates. Use the columns and the rows to guide you. Consider one gene at a time. The diagram now illustrates a 9:3 to 3:1 ratio of phenotypes among the chicks. The ratio is specifically 9 Colombian with blue egg genotype, 3 non-Colombian with blue egg genotype, 3 Colombian with a white egg genotype, and 1 non-Colombian with a white egg genotype. This is the usual ratio expected when mating together chickens heterozygous for 2 particular characters. It works as long as the alleles are fully dominant and recessive and are not closely linked to one another on the same chromosome. For this reason, it's worth remembering the ratio. Then you won't have to draw a Punnett square out every time you are interested in a different dihybrid cross. We can think about the ratios in terms of chance. Thus, the chance of a chick exhibiting both dominant characteristics, that's to say both Colombian and the blue egg genotype, is 9/16, equivalent to 56.25%. For chicks that are non-Colombian with a white egg genotype, it is just 1/16 or 6.25% chance. The chance of chicks exhibiting one particular dominant characteristic with the other recessive characteristic is just 3/16, which is 18.75%, with a 3/16 chance of exhibiting the alternative dominant and recessive characteristics. There is one other way of thinking about this, depending on how you think about things, and that's to consider what could happen when lots of similar dihybrid crosses were made. Then 56.25% of the chicks might exhibit the dominant phenotype at both loci. 18.75 would show each of the mixes, that is, one dominant and one recessive phenotype, while just 6.25% would show both recessive phenotypes. Of course, these are the theoretical expectations. In real life, the observed percentages would be approximate. The more such crosses that were considered, the closer the real percentages would be to the theoretical ones. You should note that if we consider each tray separately, there's a 3:1 ratio of blue to white egg layers and a 3:1 ratio of Columbian to non-Columbian. This is what we've observed previously and is what you expect from mating together chickens heterozygous for any one particular trait. So it's the 3:1 ratio we see from a monohybrid cross. If you have followed this video okay, you should be able to say what the chances of a chick that is true breeding for the dominant allele of both genes. If you need to, find the Punnett square and have a look. Then drop me a comment to let me know that you have worked it out. Then you will find that there is a new video telling you how many chicks you would need to breed to be sure of getting chicks with rarer genotypes, such as those expected to make up 1/16 of chicks in a hatch. So, things to remember about dihybrid crosses. Firstly, mating together chickens that are heterozygous for any two particular characters of interest is to make a dihybrid cross. Dihybrid crosses result in an expected 9:3 to 3:1 ratio. Approximately 9/16 of the chicks are expected to be of both dominant phenotypes. 3/16 have one of the dominant phenotypes only, with 3/16 having the alternative one, and 1/16 is expected to show both recessive phenotypes. If only a single dihybrid cross is made, then the 9:3 to 3:1 ratio can be thought of in terms of chance. For example, there being 56.25% chance of the double dominant phenotype occurring in a chick and a 6.25% chance of the double recessive phenotype.